All right, in chapter 24, we'll be looking at the urinary system, which includes the kidneys, the ureter, the urinary bladder, the urethra, and we'll be talking about the physiology, the functioning, and a little of the anatomy for the kidneys. So let's begin by looking at just an overview of the urinary system, and there's two main functions seen here. One is excretion, and then the other one is regulation of various things. So let's look at excretion first. There's really three main parts to excretion, and we'll spend the majority of our time in this chapter talking about these three processes: filtration, reabsorption, and secretion. And then the kidneys also help in regulating various things, including the different ions that you have. And there's another chapter we'll look at electrolytes a little more closely, but ions or electrolytes that you have in the body, these are the most common listed out for you: sodium, chloride ions, potassium ions, calcium ions, bicarbonate ions, and then hydrogen phosphate ions. These are all part of your electrolytes in the body, and it's one of the roles of the kidneys is to help balance the concentration that you have for these different ions. Another very important ion that you have is hydrogen ion, and so by balancing, your kidneys have the ability to balance your hydrogen ions. That helps to balance your pH levels, and so we'll talk about that a little bit. Your kidneys, of course, are balancing body fluids, and because of that, that's connected to your blood volume, and as your blood volume changes, that also affects your blood pressure. The kidneys produce a hormone, and that's called erythropoietin. That was mentioned briefly back in chapter 19, but erythropoietin is a hormone that helps in the process of blood cell erythropoiesis or blood cell synthesis, and then it helps in the process to make vitamin D as well. So let's look briefly at a few anatomical terms, and then we'll look at some of the microanatomy of the kidneys. First of all, these are retroperitoneal. That's a term that was first covered back in chapter 1. It just means that it's behind the peritoneum membrane. It's a serous membrane in the abdominal cavity, and this actually is behind that. The kidneys are relatively bean-shaped, roughly the size of your fist, and you can kind of see from the diagram there, they're located between the 12th thoracic vertebra and the 3rd lumbar vertebra. Is kind of where their location is. They are connected through some ligaments to kind of suspend them in the abdominal cavity. Now let's look closer at the microanatomy. You'll be covering the gross anatomy or the macroanatomy in the lab portion of the course, and so we'll be doing a dissection, but here you're going to be seeing the microanatomy. First of all, make sure you know this term, the nephron. The nephron is the functional and histological unit of the kidney. It's the smallest part of the kidney. It is microscopic. Each kidney would have millions of these, but if you can imagine, the smallest part that has everything that it would need to function. And so those are really four main parts to a nephron. That includes the renal corpuscle, the proximal tubule, the loop of Henle, and the distal tubule. Now, this chapter can be a little challenging. What you'll need to do to kind of help you through the chapter is to start learning the vocabulary, because some of the terms you may not have seen before, and there's just a lot of vocabulary terms that you need to start memorizing and studying. The nephron for one, and then the different parts. So in this picture, they've kind of outlined a little section, and they show you really two nephrons, and based on the definition of the nephron, it includes a renal corpuscle, which we'll talk about, which is located here, and then you've got a set of tubes or tubules, beginning with the proximal tube, and then it goes down and up. This is the loop of Henle, and then back up, and then you have a distal tube as well. So let's look at this renal corpuscle. The renal corpuscle is really made out of two sections, and so that's going to be the glomerulus and the Bowman's capsule. The glomerulus is a set of capillaries where the filtration process takes place. And so in this diagram, you see kind of a cross-section of the Bowman's capsule, or you see a cross-section of the renal corpuscle. The Bowman's capsule is what's on the outside of it, and then on the inside you see the glomerulus. All of the little capillaries inside are called glomerulus, or sometimes they're just called the glomerular capillaries. This is where filtration is taking place, And so notice that you have an afferent arteriole. Afferent means it's going toward the structure. Efferent means it's coming out. And so you have a small artery or arteriole taking blood into the renal corpuscle for filtration. That blood is going to be filtered here, and then it's going to pass through the efferent arteriole coming back out. The Bowman's capsule, this is on the outside. This is the outer part of the renal corpuscle. There's two layers to this, and they use the terms associated with membranes a lot of times, parietal and visceral. Parietal is the outermost layer, and the visceral is the inner layer. But let's look at that a little closer, because there's some interesting things that happen here with the Bowman's capsule. First of all, the parietal layer, that's just an outer capsule layer. It's made out of simple squamous epithelial cells, and so you can kind of see that in this diagram here. So you're very thin, relatively thin, flat epithelial cells on the outside. And then the inner part of the Bowman's capsule is what's called the visceral layer, and that is made out of specialized cells called podocytes. And what those podocytes do is kind of wrap around all of those glomerular capillaries on the inside. And so the visceral layer is made up of these podocytes here, and I'll show you another picture of the podocytes. They're basically cells. The prefix here, podo, refers to feet. These are cells that have foot-like extensions coming off the cell, and they kind of wrap around each of these capillaries. This is a image that shows the podocyte and the capillaries and how they're kind of wrapped around it. The cell body of the podocyte is kind of right in this area, and then it has extensions coming out that kind of entangle and wrap around all of these little capillaries. What it's doing is making a filter. It's basically creating a filter. It's kind of putting, like you're wrapping your fingers around something, and you create a little makeshift filter here, and that's going to help in the filtration process of the blood. So let's talk about that filter. There's really three main parts to creating a filter. There's the fenestrae, we've seen that before, basal lamina, and then the filtration slits based on those created by those podocytes. The fenestrae are those same openings or windows or pores that you find in the capillaries. That allows for certain things, small things, to kind of pass through, and larger things to stay in the blood. So if you're filtering your blood, you don't want to, you don't want your erythrocytes, your blood cells, to pass through, and so you need to create a filter system that allows the smallest of the particles, the waste products, the salts, the electrolytes, some of the smaller waste products, ammonia, the urea, all these types of things can pass through, but you don't want, your platelets, your white blood cells, your red blood cells, that doesn't need to come out, because you need to maintain those inside the bloodstream. So you're trying to create a very selective filtration membrane. Well, to do that, you have the fenestrae. You have a layer of called the basal lamina layer. That's just a thin matrix layer. You can kind of see it here on the diagram. It's like the basement membrane you would find in epithelial cells, and then the filtration slits that are created by those podocytes wrapped around it. So you have a three-layer system here to try to create a membrane. Now, the other part of the nephron are the tubules. You've got three sets of tubules that make up a nephron, and these are here for the processes of reabsorption and secretion. So filtration is going to take place inside of the Bowman's capsule, inside of that renal corpuscle region, because of all the glomerular capillaries that are there. And then the processes of reabsorption and secretion, which we'll talk about a little later, those are going to be taking place inside of the renal tubules, beginning with the proximal tube. So if you kind of trace the pathway here, here's our afferent arteriole coming in. It's carrying blood into the renal corpuscle to be filtered. All the capillaries inside, that's the glomerular capillaries. Filtration is going to take place here. So filtrate is going to be created. It's going to be passing through the filtration membrane. Now you've got to take this filtrate, and you need to reabsorb a lot of that filtrate back into the bloodstream. A lot of the filtrate that's produced, most of it, actually 98, 99% of that filtrate that gets produced here has to be reabsorbed back into the bloodstream. And that takes place in these tubules. The first tubule that you come to is going to be the proximal convoluted tubule. Proximal is just a directional term. It just means closer to the point of attachment. Convoluted just means that it's kind of curved, and you can kind of see the curve in the diagram here. And so you have the proximal convoluted tubule, which is mainly simple cuboidal epithelial type cells. Notice that they have a lot of microvilli on the edge of the cells. Anytime you see microvilli, you usually think of increasing the surface area. And in this case, you're trying to increase the surface area so you have more area for reabsorption to take place. Next, you would find the loops of Henle. And again, these have a descending portion. So this diagram is a little smaller, but you go through the proximal tube and then you start going down. That's the descending loop of Henle. And then it curves back up to go through the ascending loop of Henle. And so we'll talk about briefly the reabsorption process here. You kind of have a mixture. In some areas you have simple cuboidal epithelial, so you don't have as much reabsorption there. Some places you have simple squamous epithelial. The flatter type cells allow a little bit easier reabsorption because the cells are flatter. Then lastly, you have the distal tubule, and we'll talk briefly about that a little later as well. This is a relatively short area of the tubules, mostly simple cuboidal. You don't have very much, you don't find very many of the microvilli, so it doesn't really do a lot of the absorption. There are some functions and specialized cells that we'll talk about in another video right around the distal convoluted tubule, and that helps to balance the filtrate coming in, and so we'll look at that mechanism a little later. Now, there's one other set of tubes called collecting ducts. Now, these technically are usually not part or not considered part of the nephron. The nephron is mainly the three sets of tubes and then the renal corpuscle. But here we find that there are collecting ducts, and that's where all of the different distal tubules come together. And so then they start funneling the urine from the distal tubules. Will all collect in the collecting ducts, and eventually that will lead out of the kidney and make its way to the ureter. Now, in section 3, it really starts looking in more detail at the physiology, how each of these processes are going to function, the filtration, reabsorption, and secretion. Figure 24.11 does a good job of just kind of summarizing everything. So as you're going through each of these processes, it might be good to kind of refer back to this figure sometimes, because it kind of shows you kind of an outline, a diagram of the nephron, of the different parts. You can see the renal corpuscle, the proximal tube, the distal tube, the collecting ducts, the loop of Henle, and all of that. You see these capillaries that are right through here. They play an important role in reabsorption, which we'll get to in a little while. And then the arrows show you what's taking place in these areas. So F stands for filtration, so that's the only place that filtration is really taking place here. And then R is showing you reabsorption, and notice the arrow, you're reabsorbing fluid, filtrate back into the bloodstream. That's why you have all of these capillaries and vessels right close to these tubules. And so anywhere you see the R with that arrow, it's showing you the direction of flow. So you're removing filtrate from this flow here and putting it back into circulation. And then S we'll talk about at the very end. This is secretion. You have an opportunity here for some other waste products to make their way from the blood, from the vessels here into the tubules. Notice that's going the opposite direction as the reabsorption. So it's going back into filtrate, and you have a few opportunities here for a few more waste products to kind of enter into the filtrate. And so we'll talk about that at the last part of this chapter. So let's concentrate on the first part, which is filtration. Filtration takes place inside of the renal corpuscle. It's because you've got the blood flowing into those capillaries, glomerular capillaries, and it's the pressure, the blood pressure that's basically is the driving pressure that's forcing the plasma, really, I put water up here, but it's really the plasma portion of the blood. And then, different waste products and some electrolytes and things, forcing that out into the Bowman's capsule to form what we call filtrate. one thing to note is the reason why we use the term filtrate and not urine at this point is because this filtrate has not been concentrated. This is a lot of fluid, a lot of water, and some salts, and maybe even some maybe possibly some proteins and things, and possibly some glucose and nutrients. Well, that filtrate has to be reabsorbed, or a lot of it does, so that you can reabsorb a lot of that fluid. You don't want to waste it. You need to reabsorb any proteins and sugar and glucose and nutrients that may pass through there. And so once it's concentrated, then you use the term urine. All right. So a couple of terms associated with the process of filtration. The first one is called the renal fraction. The renal fraction is the part of the proportion of your cardiac output, your total cardiac output. Now that's we looked at that briefly back in the chapter on the heart. For an average person, the cardiac output might be about 5.5 liters, 5.6 liters, or 5,600 milliliters every minute. That's how much blood is pumping out of the heart every minute. Well, the part of that goes through the kidneys is what's referred to as the renal fraction. They estimate that about 21%. So about 21%, or a fifth, of the blood that's passing, coming out of the heart, is passing through the kidneys every minute. And so if you multiply those two, 21% times 5,600, you multiply those two, you get a flow rate of about 1,176 milliliters of blood passing through the kidneys every minute. Well, if our goal really is to find out how much is really being filtered. And so really what we need to know is what part of the blood really gets filtered. Well, it's the plasma portion. The blood cells don't come out. We're really focused on, the plasma portion that's being filtered. And so to do that, the plasma, if you go back and look in chapter 19, we said that the plasma portion was about 55% of the blood is plasma. And these are just average numbers. And so if you take your renal blood flow rate, multiply that times 55%, or 0.55, then you get about 650 milliliters of plasma that gets filtered every minute. All right. So we're kind of narrowing it down a little bit. So you started out with the total volume of blood coming out of the heart, looking at the total amount of blood going into the kidneys, which is a little over a liter. And now we're looking at how much plasma is going through the kidneys each minute. It's about 650. There's one more thing we need to look at, because not all of that plasma gets actually filtered out. So, the question is, how much actually gets filtered out? Well, it's about 19%, roughly. And, I don't know how this is all measured, but roughly about 19% of the plasma that goes into the kidneys really gets filtered out. And so now we can calculate what's called the GFR. This is one of the key things about filtration, glomerular filtration rate, your GFR rate. This is how much filtrate that is actually produced by the kidneys each minute. And so to calculate that, we took the 650 milliliters of plasma that gets filtered by the goes into the kidneys each minute, multiply that by the filtration fraction, which is actually the part that gets filtered, because not everything is going to get filtered. It just kind of passes through. That's about 19%, and that comes up with 123.5 milliliters of plasma that gets filtered every minute. And so that's what's referred to as the filtrate, which is a huge volume. 123 milliliters every minute gets passed through both kidneys. So that's a total for two kidneys working together of filtrate being produced per minute. If you want to look at that on a daily basis, you could kind of do the math. I've done that for you. 1,440 minutes in a day times 125, that's our rounded up number there, 125 milliliters of plasma per minute. That gives you 180,000 milliliters a day, or 180 liters a day worth of filtrate being generated throughout the day. And so, a lot of that has to be reabsorbed. Most of it is reabsorbed during the process of the day. And so, your average urine production, of course, that changes a lot, but on average is usually between 1 to 2 liters. And so that means that 98 to 99% of that filtrate actually gets reabsorbed back into the bloodstream so that you concentrate that filtrate into what we call urine. Now, one other thing that we'll look at for filtration at this point is the filtration pressure. The driving force we said is blood pressure. Well, that's given a special name. That's called glomerular hydrostatic pressure, GHP. That's the blood pressure inside of those little capillaries inside of the renal corpuscle, inside the Bowman's capsule here. And that is an outward pressure. That's how much pressure here that's pushing that filtrate through the filtration membrane. That's the GHP. And so they give you some numbers here on the diagram. They say, well, you know, GHP might be somewhere around 50 millimeters of mercury. It's the same units that we measure blood pressure in. This is a blood pressure. And so it's hydrostatic pressure pushing the filtrate out to create, pushing the plasma out to create the filtrate. But you do have two other pressures that are associated with filtration pressure. The next one is called glomerular colloid osmotic pressure. Osmotic pressure is kind of an inward pressure. It's because you have some proteins already in the fluid. So whenever you're looking here, you've got fluid on the inside of the vessels, inside the capillaries, and there's fluid out here in the Bowman's capsule and out here in the tube. Well, that fluid out here has some proteins associated with it, and that creates an osmotic type pressure. And so what that is an inward pressure. They estimate that on average, and it changes to be about 30 millimeters of mercury. And so you have two pressures kind of going in opposite directions. Last pressure is called capsular hydrostatic pressure. Again, this is an inward pressure as well. This is here just because you have that fluid on the outside, it's kind of pushing against these vessels. It's just a little water pressure. That's why it's called hydrostatic pressure. It's called capsular because it's the pressure of the fluid inside the capsule, and it's just pushing against these capillaries. So that's an inward pressure, and it's they estimate it to be about 10 millimeters of mercury. And so if you wanted to calculate your overall net filtration pressure, you would have to take your GHP and subtract off the other two pressures. So take GHP, which is 50, subtract off 30, subtract off 10, and you will be left with the net, which is 10. Notice it's a positive number, which means that fluid is still, it's a positive number, so it means that fluid is going to be wanting to go out or leave the capillary. The plasma will leave the capillary and be forced out into that space inside the capsule, the glomerular capsule or the Bowman's capsule.